Hi, my name's Paul Tyler. I'm a primary school teacher and primary science specialist. Before going into teaching, I was a biochemist and research scientist. I'm going to talk to you today about the superbug battle and some of the research that I did as a scientist. So a little bit about me first and about my inspiration. Started at primary school, my interest in science, and you can see from the image here, I was experimenting with the effect of gravity on eggs, how much hair you can see through, and also how collar size affects your flying ability. I then got a bit more serious, and my older brother was very much an inspiration to me. He was into his chemistry and had all sorts of chemistry sets, and I used to spend hours in his bedroom doing chemistry with him. I was then very lucky and went to St John's School in Leatherhead, the picture at the bottom, and the science department there were an absolute inspiration to me. I had several superb science teachers. A lot of the roofs you can see in the picture are also copper, and I was also always fascinated by the oxidation and carbonation of the copper to go that kind of aquamarine color. I did A-levels in chemistry, biology, physics and maths at St John's, and it's testament to the quality of the teachers that they managed to get me through those. I then disappeared off to Leicester University, where I studied biochemistry, and I was very, very lucky at Leicester to have some superb lecturers and tutors, including Professor Sir Alec Jeffreys, who was my genetics lecturer and tutor. Professor Sir Alec Jeffreys was the founder, the inventor of DNA fingerprinting, which is used all over the world now. Dr. Annette Cashmore, you can see at the top there, was one of my genetics lecturers and tutors as well, who sadly died a few years ago. But Annette was an absolute inspiration to me. And Dr. Colin Jones, who was my biochemistry lecturer and also my third year project supervisor. And Colin and I had a fantastic relationship, and he had a lot of patience with me during my time there. Having left Leicester University, I ended up working for a startup biotech company called Pantherix, which was founded by Professor Bill Shaw and Dr. Bill Primrose, and in 1998. And I was the first employee of the company and arrived when the company was still based at Leicester University. Right at the start of the biotech boom, and we were a structure-based antibiotic design company, and we were looking at pantothenate kinase as one of our targets. You can see in the bottom corner there, that's pantothenate kinase for Mycobacterium tuberculosis. The company, after a year or so, moved up to Glasgow, and we grew rapidly. Up to a maximum. At one point, we had 60 scientists working in this incredible custom-built facility. In about 2003, 2004, the biotech bubble burst, and funding for biotech companies basically ran out. However, when the company was taken over by AstraZeneca, who bought over all the intellectual property and several of the employees, I had kind of fallen in love with Glasgow and decided to stay. So it became time for a bit of a career change. I went and worked for 3 years for Scottish Rugby as a development officer and high performance coach, coaching and working in primary schools. And that kind of made me realize that working with young children at that really impressionable age was what I wanted to do. So I went back to college. I did my PGDE at Strathclyde University at Jordan Hill College that you can see here. And I'm currently teaching in Kirkhill Primary School, which is a large 4-stream primary school, just over 700 pupils, just on the south side of Glasgow. And hopefully, one of the things that I see myself as doing is inspiring the next generation of scientists who will hopefully be around to change the world for the better. One of the things that really got me inspired when I was at university was the opportunity to get my first published paper. And I'm quite proud to say I have several published papers in various fields. This was my third year university project, and I spent my summer holiday. I enjoyed it so much. I spent my whole summer holiday up in Leicester working on it. I had an amazing mentor in Dr. Colin Jones, who you can see as the one of the authors on the paper. And this all came from my fascination with bacteria and enzymes. And I was really keen. This was one of the most sought after projects in third year, and I was delighted that Dr. Jones saw something in me. And this really sort of sparked the next phase of my career, and made me want to go on and study enzymes particularly, and bacterial enzymes even further. So what's a bacteria? Well, a bacteria is a single-celled organism. There's around about 30,000 distinct named species on Earth, and they're found in every ecosystem all over our planet. Us humans, we are basically huge bacterial cultures. We have between 40 and 80 trillion bacterial cells on us and in us at any one time. And we all have a unique microbiome, which is as unique as our fingerprints or as our DNA. And this is actually used, our microbiome can now be used by forensic scientists for crime scene analysis. Our bacteria are essential. They act as part of our immune systems. They digest foods such as dairy that we can't break down otherwise. They provide us with nutrients, vitamins, minerals that we need to survive. We've also exploited bacteria in a number of ways. So we use them to create products such as enzymes, such as ethanol, make drugs like antibiotics and vaccines, make biogas such as methane. We use them to clean up oil spills and toxic wastes. We use them to clean plant pests. We use them in gene therapy. We use them to ferment foods. They're used all over the place, and they're incredibly useful. So how are they different from us? How is the anatomy of a bacterium cell different from a human cell? Well, humans are multicellular organisms. We have about 37 trillion cells in an average adult. And there are similarities between our cells and the single cells in a bacteria. They both have DNA. They both have things called ribosomes, which we'll talk about a little bit later. They both have a cell membrane, but more importantly, there are some significant differences. Bacteria have a cell wall that surrounds the cell, which human cells don't have. They also don't have mitochondria, and they don't have a nucleus. Everything in a bacterial cell is kind of floating around in its cytoplasm, whereas a human cell is much more organized with this nucleus. There are also lots of differences at the metabolic level, and later on we'll see just how important these metabolic differences are. So why do we want to kill some bacteria? Well, we couldn't survive without the bacteria that live in us, in our gut, in our intestines, and also live on us. However, there are a significant number of bacteria, as you can see on this list, which are harmful to humans, and these are called pathogens or pathogenic bacteria, and they cause some pretty serious diseases, things like tuberculosis, typhus, the plague, diphtheria, typhoid, Legionnaire's disease, cholera, dysentery, and pneumonia. And these diseases caused by these pathogenic bacteria kill millions of people around the world every year. And this is why we need to be able to selectively kill specific bacteria, hopefully without harming the bacteria that we need in order to survive. So how do we kill bacteria? Well, the best thing we can do is not let them into our bodies in the first place. And we can do this by purifying water and making sure that the water we drink is clean. We can do this by having effective waste systems for our sewage. We can do this by good hand hygiene, by washing our hands after specific activities. We can do it by cleaning surfaces around where we work and around our homes. We can do this through vaccinations. And then if we do become infected and get ill, we can use antibiotics. And on the right-hand side here, you can see penicillin, which is one of the most famous antibiotics, and penicillin and its derivatives are still the most widely used group of antibiotics that we have. So how do antibiotics work? Well, remember earlier we talked about the differences between bacterial cells and human cells, and that's what antibiotics target. They target those differences, and there's four main ways that they can act. They can target the cell wall, and if you remember, human cells don't have a cell wall. So if you're targeting the cell wall of a bacteria, you're not affecting human cells. They can block DNA synthesis within the cell. DNA synthesis inside a bacterial cell is a slightly different process to it is in a human cell, and so that can be targeted. They can prevent RNA being produced from DNA. Now, RNA is really important. DNA is deoxyribose nucleic acid, D meaning 2, and RNA is just ribose nucleic acid. And the difference between them is that RNA is then used to produce proteins, to synthesize proteins. So if we can block RNA being produced from DNA, we can block proteins being made. And then the last step is to attack the ribosome that you can see there and stop essential proteins being made in the cell. And again, bacterial ribosomes are different enough from human ribosomes that they can be targeted selectively. Antibiotics are just chemicals that kill bacteria and hopefully have little or no effect on humans. Penicillin was the first antibiotic discovered by Sir Alexander Fleming, and it's a naturally occurring chemical secreted by mold to kill bacteria. And since its discovery, it's been altered in lots of ways to produce a whole family of antibiotics with the same core structure. And you can see the image on the screen, the bit in the top left-hand corner, the variable group, that's the bit that gets changed in different penicillin family antibiotics. So one of the issues that we have at the moment is that antibiotics are becoming less effective around the world. And so why is that? Well, since the discovery of penicillin, the use of antibiotics has grown rapidly to the situation now where they're very commonly overprescribed by doctors, even when they're not needed, even when there's not a bacterial infection. They're even added into animal feed for farm animals. And those eventually make their way into human populations. The overuse of antibiotics, coupled with the fact that bacteria multiply very quickly and they adapt and mutate very, very rapidly. And this has led to the rise of something that the media like to call superbugs. Things like MRSA, methicillin-resistant Staphylococcus aureus, MRSA, which is resistant to almost all antibiotics that we have. The concern is that if we continue to use antibiotics at the rate we are just now, it's not going to be very long before they're all useless. MRSA can be grown on agar plates containing really high levels now of antibiotics. And this is MRSA in the image, methicillin-resistant Staphylococcus aureus, being grown on an agar plate containing antibiotics. And you can see as the concentration from the bottom right-hand corner, the concentration of antibiotics goes up, you can see there's less bacteria being grown, but there are still colonies of bacteria there. And these colonies are resistant to the antibiotics, and they will be able to multiply and be resistant wherever they are. This is a real problem, and a huge amount of scientific expertise and money is currently being invested to find solutions. So what can we do? Well, the research that I was involved with in the late 1990s and early 2000s was looking at possible new antibiotics that target bacteria in a very different way from the 4 ways that we've discussed. At Pantherix, we had a multidisciplinary group of scientists looking at these differences between bacterial cells and human cells that were quite well categorized by scientists up to then that we could exploit. At one point, there were 60 of us in the team, including biologists, geneticists, biochemists, chemists, microbiologists, crystallographers, all working on the challenge from slightly different angles and trying to find a solution. What we were studying was the metabolic pathways that all living things have. This is a map of the metabolic pathways that humans have in order to survive. So this is a map of every chemical reaction that has to take place in your body in order for you to survive. These metabolic pathways are a very tightly regulated series of chemical reactions that break our food and our nutrients down into useful things that our body can use. So if we have a look at one particular one here, this is the metabolic pathway for glucose, which I'm sure you'll know is sugar. And it shows you what happens to glucose when we eat it. And each step of the pathway, coming down from glucose through glycolysis to pyruvate to acetyl coenzyme A, and then into this thing called the Krebs cycle, every step needs an enzyme. And you can see the enzymes there in red, and they have slightly funny names, things like glyceraldehyde 3-phosphate dehydrogenase and pyruvate dehydrogenase. But the names explain to scientists, to biochemists and chemists, what those enzymes do and what the action that they have is. And these enzymes are essential for each step. Bacteria have metabolic pathways as well. And this particular metabolic pathway here, the metabolic pathway that takes pantothenate all the way through to coenzyme A at the bottom, was of particular interest to us. And this is because this metabolic pathway was different enough in bacteria to humans for us to think that we would be able to target it without affecting humans. So coenzyme A is essential in humans and bacteria, but they both make it in slightly different ways. And the enzymes involved are sufficiently different for us to think we could target them without affecting the human host. Once we'd identified this pathway as a possible target, we had to identify which of the enzymes were going to be the best ones to target. You can see a series there of 5 enzymes going through this metabolic pathway. 2 of the enzymes stood out, and they were pantothenate kinase, which is the first step, taking pantothenate to 4-phosphopantothenate, and the 4th step, which was phosphopantethine adenyl transferase, PPAT. These 2 looked to be the best because these were what we call the rate limiting steps. So these determined how fast this reaction happened through the pathway. They were also quite complex steps. And also different enough from the steps in a human. And the plan was to find a chemical or a drug that would target one of these enzymes very specifically, block this pathway in bacteria, stop the bacteria making coenzyme A, which would kill the bacteria without affecting the human host. So we needed to look into how enzymes work. And we've known for a long time how enzymes work. They're biological catalysts, and they facilitate and speed up chemical reactions within living cells. And every step on that metabolic map we saw involves enzymes to make the chemical reaction work. They're very specific enzymes. They only do one job. It's like a lock and a key, where only one key will fit a lock, and you have to have the right key. An enzyme can't go away and do another job anywhere else. It has that specific job. Enzymes have an active site, which is where the chemicals, the substrate in this case, you can see that in green on the left there, binds. And this forms what's called an enzyme substrate complex. And at this point, the substrate and the enzyme are quite tightly bound together. This is when the chemical reaction happens. And in this case, the products complex, you've got the enzyme products complex, the products are formed, and the enzyme then releases those products for the next enzyme in the pathway to work on, leaving the enzyme free for the next substrate to move in. It's this fact that enzymes are so specific for just one chemical that makes them very good targets for antibiotics, because if the antibiotic can mimic the substrate or the product and bind tightly into that active site, it'll stop the enzyme working, and the metabolic pathway will be blocked, and that will kill the bacterial cell. So we identified pantothenate kinase and phosphopantethine adenyl transferase as targets for antibiotic design. And what we needed to do then is we needed to make and purify large quantities of these enzymes to work with. First thing we did is our team of geneticists cloned the gene that encodes for the enzyme the enzymes that we were interested in. And we used E. coli bacteria to make lots and lots of the enzymes. We then broke the bacteria open, we smashed them open, and released all the enzymes and all the other proteins from inside the bacteria. We then had to purify the ones that we wanted. Now, this machine here is called an FPLC, fast protein liquid chromatography. And this allows us to pass the contents of the bacteria through a series of special columns and separate them based on things like their size, their chemical composition, their charge. If you imagine a mixture, and you can try this at home, a mixture of sand and salt, and maybe something like iron filings or paper clips. The iron filings can be removed from the mixture using a magnet because they're magnetic. The salt can be dissolved in water and then the sand can be filtered out. And the salt can then be got back by evaporating the water. So you end up with each component, the salt, the sand, and the iron, either the iron filings or the paper clips, completely separated and pure from all the others. I spent pretty much all day, every day of my research career working with one of these machines and purifying enzymes. Had to be done very, very carefully, because the pure enzyme had to be, at the end of it, had to be perfectly folded still, and it had to be active. So it could still carry out its chemical reaction. So you couldn't do anything too violent to these enzymes, because that would break them. Every single thing that we did in the laboratory had to be documented in a handwritten lab book. And in my 7 years of research, I filled over 20 A4 lab books, each containing about 100 pages. At each step along the way, we had to see how pure the enzyme, the target enzyme that we wanted was. The average enzyme purification took 4 or 5 steps, after which it was then pure enough to work with. And we used something called gel electrophoresis. And what we did is we loaded the enzymes, the mixtures in one end of this gel, which is a bit like a jelly, and then we passed an electric current through it, and the enzymes very slowly moved through. And they all stopped at particular places. Here you can see a gel showing a set of size markers down the left-hand side, and then each stage of the purification going from left to right. So the first column is the broken open cells. So that's all the proteins and enzymes from those cells. The next stage shows a wash step, and you can see there's not really very much difference, but that washes away all the bits of cell membrane and cell wall that we don't want. Then steps 3, 4, and 5 are 3 different separating columns, and you can see through 3, 4, and 5 how our enzyme that we want, which is that thick band with the arrow pointing to it, gets purer and purer and purer, until in step 5 there are no other proteins or enzymes at all. At each step, the enzymes had to be tested to check that they were still working. Once the enzyme was pure, the next step was to grow crystals of the enzyme. Now, again, you can do something similar at home by growing sugar or salt crystals, where you would dissolve lots of sugar or salt into a solution of water, to make a solution, and then you can leave it on the windowsill to evaporate the water off, and you'll get lovely crystals of salt or sugar. And we can do the same with enzymes. The enzyme was very, very carefully concentrated, and then the liquid it was dissolved in was allowed to really slowly evaporate. You can see this in the top left-hand corner. So at the top of the left-hand corner, at the top of that pot on the lid, is a little drop of the concentrated enzyme. And you can see tiny little crystals starting to form as the liquid in there evaporates. And these crystals formed. And we did this using lots and lots of different situations, lots of different liquids to try and evaporate it as slowly as possible to get the largest and the most perfect possible crystals. The crystals were then placed in the path of very strong X-rays, and a diffraction pattern was collected by the detector. From that diffraction pattern, a density map could be produced, and then there was a lot of hard work and refining to get a beautiful 3D image of the enzyme. The crystallographers that I worked with were definitely part scientist, part artist, and I wouldn't be surprised if there was quite a bit of magic involved in their work as well, because what they were able to do was to take my purified, concentrated enzymes that I'd grown these beautiful crystals from and produce something like this. So this is the crystal structure of phosphopantotheniate adenyl transferase from E. coli. And from the structure, we're able to see all sorts of things. We're able to see how the protein or how the enzyme folds up. We can see different domains of the enzyme, different bits that join it together. We're also able to see where the active site is. And if you remember earlier, the active site is where the substrate binds and then the products are released. We're also able to see right down to see which amino acids, and all proteins and all enzymes are made up of 20 amino acids. So we can see which amino acids are directly involved in the chemical reaction. This means the chemists can work out exactly how the chemicals bind in the active site, and we can calculate which bonds are going to be the best ones to target with our antibiotics. There are 3D structures for a wide variety of proteins and enzymes that are now fairly readily available. There's libraries of these online, and they've been integral to us moving forward in our understanding of how enzymes work and how our metabolic pathways, both in humans and bacteria, work. So with the high resolution crystal structure, we're able to see exactly how the chemical reaction that this enzyme catalyzes occurs. And then we were able to begin designing chemicals which would bind to the enzyme and to try and stop it working. And this is where our team of chemists took over. The key with all drugs for human use is to design a chemical that will bind to the active site of the enzyme, stop it working, but is so specific that it wouldn't harm any other enzymes in the human host. There are plenty of chemicals out there that would kill all bacterial enzymes. The trouble is they would also kill or stop working all human enzymes, and that would kill the human. So they have to be really specific. Beyond that, there's further issues to think about, even before you get to the stage of doing testing and clinical trials and things. That's to think about things like how are you going to deliver the antibiotic in the most effective way, and how can you minimize any side effects. So that's where my story finishes. At that point, Pantherix, who I was working for, we had three chemicals that were kind of ready for preclinical trials, but preclinical trials and clinical trials are incredibly expensive, and it's a very complex process to go through to make sure that any drugs that get on the market are perfectly safe for human beings to use. And so at that point, we ran out of money. The funding stopped, and the whole company was taken over by AstraZeneca. All my lab books were taken away, and all the intellectual property from the company was taken away, and is now down with AstraZeneca. But the superbug battle goes on. Epidemiologists, microbiologists are warning us just now that by 2050, superbugs could be killing 10 million people a year. And this is why antibiotic research and antibacterial research is one of the most funded areas in medical science. In 2016, a group of the world's leading experts on microbiology, virology, medicine, biochemistry, genetics, epidemiology came together to evaluate all the possible approaches which we can use to fight resistant bacterial pathogens in the future. And they decided between them on a shortlist of 7 approaches that they recommended were the most likely to succeed and therefore the best ones to be funded. And these were things like antibody therapies, like probiotics and lysins, which were all considered. Potentially the most exciting is the use of bacteriophages. Now, bacteriophages are viruses for bacteria. And I realized just now the word virus is a bit of a dirty word, but these bacteriophages, these bacterial viruses are incredible. They're very, very specific to certain species of bacteria, and every species of bacteria has a set of bacteriophages that infect it. In almost all cases, they cause the rapid death of the bacteria and can stop any bacterial infection. They've already been used in some countries to treat bacterial infections for decades, and the idea is now making a comeback in the wider scientific and medical circles. And it's just another beautiful example of the power of biology to cause and cure infection and illness. So thank you very much for your time today. I hope you found this interesting. Possibly it's inspired you to ask some more questions and do some research on your own into superbugs and the ongoing fight against them. Thank you very much.